I would like to welcome our speaker today. It's Professor Dr. Carsten Grashoff, currently a group leader at Institute of Integrative Cell Biology and Physiology at University of Münster in Germany. He started his career in Germany up to his PhD studies, then moved as a research associate to University of Virginia, USA, for a few years, and then went back to Germany to the Max Planck Institute, where he started his own research group to finally, in 2018, start at University of Münster, where he has been ever since. So a bit of background about his current research. His group is interested in mechanical forces and signals and how distinct proteins experience these mechanical forces inside the cell. They have developed a microscopy-based technique and novel optomechanic tools to modulate molecular linkages in cells and are using them to define the molecular details of mechanobiological processes with a special focus on cell adhesion. So we are looking forward to your seminar. If anyone has a question, you can write it in the chat and then we will read it at the end of the talk. Or when the talk is over, you can raise your hand in the Zoom and ask it yourself. So please, Professor, if you could share your slides. The screen is yours.
Nuria, thank you very much. Let's see whether that works. Can you see this? Can you see the slides?
Not yet.
Not yet.
Sure. Now I know why. I first have to share it. Yeah. Okay. How's this?
It's coming now.
Yes, here we are.
Okay. Well, thank you very much, Nuria, for the kind invitation, first of all, and the introduction now. First time you wrote me, I was not quite sure whether I fit to the seminar series because our expertise is not really on intermediate filaments. But then on second thought, I realized, yeah, maybe that could be in some ways interesting in what we have done so far, also to your community. And so I try to assemble a talk where maybe you guys can benefit a little bit from what we are doing. And I'm looking forward to an interesting discussion. So you rightfully said that we are interested in cell mechanics. So in principle, we would like to understand how cells are able to process mechanical signals. Yeah? And the process that we are very much interested in, it's not the only process that we are working on in the lab, but the major focus is cell adhesion. Yeah? And here you can see already a cell doing cell adhesion. And I don't want to waste too much time on my introduction, but rather go into the data because I think you all are aware why mechanobiology and cell mechanics is important, right? And so I have just basically one introductory slide that you can see here, and it's all about how mechanical signals are processed in our cells, in our tissues on molecular scales. And these molecular scales, I have to emphasize a little bit because this is all what our experiments are about. So a lot of these processes that I will be talking about well understood on conceptually, on a structural level, on the complex level, let's say, but how the molecules in these structures that I will talk about in a second, how they really contribute to force processing, this is often not so clear. So the exemplary tissue that I often show is shown here. Let's say we look at an epithelium, and I'm sure many of you in this EF community are looking at epithelia as well. Well, if you look into the into such structures, such tissues, you will find that there are subcellular complexes in there that hold this tissue together and provide mechanical stability. This is, for example, adherence junctions, focal adhesions, also desmosomes, hemidesmosomes, and these adhesion structures, we know this, are important for the mechanical integrity of the overall tissue. I mean, that's clear. We also know from genetic studies, if you delete those structures, the mechanical integrity will be heavily impaired. And we also know what kind of molecules are in these structures. Yeah? So for the cell adhesions, of course, these are cadherin-based receptors. And for the cell matrix adhesions, these are integrin-based receptors. And these receptors connect to the cytoskeleton through individual proteins. Yeah? And I already told you that we think we have a good understanding of why we need these complexes, but I don't think that our understanding of what these molecules in these structures do is very well developed. So what we typically are looking at are these focal adhesions, these integrin-based structures, but we have also worked a little bit on desmosomal structures, and this is where I can make that link to your community. So I try to talk a little bit about the actin-based adhesion structures, because based on these structures I can nicely explain how our experiments work, what the rationale behind this experiment is, and then I will try to link that to desmosomes. And in fact, I think that you have to look at both if you want to understand the tissue, because all these structures are there. Often the individual communities look at them in isolation, but mechanically speaking, they're of course all interconnected. Yeah, and this is also what I think one of my take-home messages will be, that these structures make different things, but they are not working in isolation. They are somewhat complementary to one another. Okay, and so I already showed you here one reason why we think we can make a contribution is that we think we need technologies that allow us to look at the molecular scale details of these processes. So technologies that allow us to look which molecules in these subcellular structures are important, for example, for a force transduction process, and which ones maybe not. Okay, as indicated already, I will start with talking about cell matrix adhesions. I apologize, I have to go back to papers that are published for quite a while, but nevertheless, I think it's important to lay out the overall technology that we had developed at the time, and then later on I will talk about the desmosomes and desmoglacanons. Okay, so these are the structures I will be starting with, the focal adhesions. These are the structures that are actin-based, and they allow cells through integrin-mediated adhesion to attach to substrates, right? And here in this picture, you see the focal adhesions in this pinkish color, and they connect to the actin cytoskeleton that's shown here in this green color. And we know that these structures are absolutely essential for not only the cell to adhere to the substrate, but also to sense mechanical signals and to transduce or transmit these signals into and out of the cell. So it's a bidirectional structure that mediates bidirectional force transduction. And when you look into this structure with a higher resolution, and this picture here, many of you probably have seen, it's coming out of a paper from Claire Waterman, where they tried with super resolution microscopy to actually see how these proteins organize in the structure. You can see there are many proteins. That's the only thing you have to take away from this picture. So it's not that easy when you look into these individual structures to find out which molecule is actually important, because often these are multimolecular structures, and many proteins could contribute to a given force transmitting process. So here at the time we wondered, okay, which of these molecules, you see the integrins in red on the outside of the plasma membrane, and you see the actomyosin network on the inside in this bluish color, and you see they're connected by many of these focal adhesion proteins, and some of them I will mention in a second. So the question is, if you have such a structure, and the same questions you can probably apply to whatever structure you are working with, the question is, how is mechanical information actually transduced across such a structure on the molecular level? Meaning, which proteins are actually transmitting the force that you think is transmitted across a structure? That would be the first question. And if you have identified such a molecular linkage, then you could also ask, how relevant is it actually that you have that linkage? Because just because it's exposed to a force doesn't mean that it's very important. At least it's not easily connected to a physiological function. So these are the two questions that I will keep throughout my talk, and come back to these questions again, because I think it helps us understand why we do our experiments. So I will start with the first one, which is very easy to understand. The question is, if you have such a structure, how do you find out which molecules are actually transmitting mechanical forces and which ones are there and have actually nothing to do with force transmission? Now, this question is, or was at the time, not so easy to answer, because the forces that you have across individual molecules are really quite small. They're in the piconewton range, so that's a billionth of a newton, and actually they're in the low piconewton range. So this is actually not easy to measure, and so at the time we had to develop a technique that allows us to monitor such small forces. And the technique is in principle easy to understand. It's something like this, a spring scale that you know from physics class. And the idea was, let's just generate such a small spring scale, make that a nano spring scale, and insert that into the proteins that we want to understand. So if you have the hypothesis, there's protein X that transmits force, let's take such a spring scale, insert it into the protein of our interest, and see whether we can see any tension in this protein. And the way the spring scale looks like is this. So it uses a biosensor that is FRET-based. So FRET is first resonance energy transfer. And as you probably know, this energy transfer between two fluorophores is always high when these two fluorophores are close to one another. And they're slow when these fluorophores are separated. And the idea was to connect these two fluorophores with an elastic linker that can be stretched by the forces that we want to measure. Well, that can, in our case, be stretched by piconewton forces. And the idea was, okay, if we find something that is flexible and elastic, that means that extends under force, but also importantly recoils when the force is over, right? Or force is not there anymore. Then we have a sensor that can probably tell us something about the force that we have by inversely correlating the FRET rate. Oh, someone is bored already. Okay. Good. So this was the idea, and since this is not new, I will make it quick. So these are these biosensors that we have developed more than 15 years ago, or published 14 years ago. And the important thing, this is I would like to emphasize, is this one here. There is not only one biosensor, but many have been generated. So if you are interested in this sort of technique, you want to make sure that you use a biosensor that fits your, experimental needs. And we have made 4 of these biosensors that cover force ranges in the low piconewton range, but they are all somewhat different in their force sensitivity. The most important thing is, if you use a biosensor that is calibrated, because it's easy to clone these things and it's easy to say what they should measure, but it's actually quite hard to prove that they really measure what you think they should measure. So our biosensors are all calibrated. We know they extend under the force that we write here, and we know they are reversible, so they also recoil, and they're actually hysteresis free. That means the extension pathways and the return pathways are exactly the same. So whenever you measure a FRET value, you can correlate that to single force. Okay, and I think people use this not enough, because you can use these different biosensors also to actually control your experiments. Yeah? Okay, so we have inserted that into different proteins. So I guess by and large you have to believe me that in principle this can work, that you can use such a biosensor, insert it into proteins, and then measure along your proteins in cells how high these forces along the protein are. And this does not only work in cells or in cell culture, it also works in animals, and we have done it in flies, but others have done it in frogs and C. elegans and mice even. So the good thing is this technique has been used by others, so it seemed to work to some extent. Okay, so I just quickly want to explain to you that we're all on the same page how it in principle looks like in case people would like to use this technique. You have to know some things that you have to pay attention to. And if you really want to do it, my advice would be just contact me and we talk about it. Because it's actually a little bit more difficult than it looks like in my talks, right? Because you don't have really time to talk about all the problems. You rather like to show how nice data you get. But it's not a simple experiment, right? Because in case of this example here, so we looked at taline, and this is one of these proteins that connects the integrins to the actin cytoskeleton. At the time, we were very interested in that because taline was known to be an integrin activator. So it binds integrin receptors directly and activates them. And that's why this taline protein was famous already before we started working on it, because if you don't have it, cells cannot adhere because they cannot activate integrins. Okay, but as you can see here from my drawing, taline is a large molecule that has more than just this integrin binding site. It has these actin binding sites and also a lot of vinculin binding sites. Vinculin is another protein that you find in these adhesion structures. and so the idea was maybe tailin is not only a protein that activates integrins, but it also connects the integrins mechanically to the actin cytoskeleton. That this is one of these proteins that is transmitting force from the integrin into the cell. And so what we did is inserting that biosensor that I just explained on the slide before into the tailin protein. And here you can see already the first problem, right? Because if you do that, you have to really insert it into the protein. C-terminal attacking or so would of course not help, because then nothing can pull on your biosensor. You really have to find a way to insert the molecule into your protein. And that's not trivial, right? So to the very least, you have to control very well whether this actually works. And we had cases where we tried to do it and it just never really worked well. And if it doesn't work, well, you don't have to measure it, because then you will measure some values, but they don't tell you much about the physiologically relevant process. Okay, in case of tailin, we are quite convinced that it actually can be a more or less functional tailin when we insert a biosensor into this protein. You have to, because we performed experiments that showed us that this tailin is still functional. And for tailin, it's actually not that hard, because if you knock out tailin, I told you, integrins are not activated, so your phenotype is quite severe. You see here this tailin 1, tailin 2 knockout cells. So these cells have no tailin anymore. They cannot adhere, they cannot spread. And then you can ask, if I put back a normal tailin that just have a fluorescent tag at the C-terminus, or my tension sensor tailin, are these cells then normal? Is the signaling normal? Is the spreading normal? Is the morphology normal? We looked at all that. This is just one example. And we are quite confident that the molecule behaves as wild type. You can measure turnover with fluorescence recovery after photobleaching experiments. And of course, we have whole flies where we replace the endogenous tailin gene with a tension sensor tailin gene, and these flies are completely normal. And it's known from flies that if you mess with tailin, then typically your fly is dead. Yeah. So here, in case of tailin, we spent a lot of time making sure that this protein behaves normal after we insert our tension sensor, and then you can perform experiments. And I will make this very short because all this is published. So the take-home message of all this is that tailin, yes, it is a protein that is under mechanical forces, that experiences forces when cells adhere, and by using different tension sensors, we can actually map these forces quite well. So we think they're around 7 to 10 piconewton that an individual tailin molecule can experience in a cell adhesion site. This data next to it is just for you to get you an impression of how all these experiments look like. We use fluorescence lifetime microscopy to quantify this data. So this is my strong recommendation. You can also use ratiometric FRET, but then you have to be very careful in analyzing your data. The advantage of doing FLIM is that you can always compare this data to any other FLIM setup in the world, and because it's a quantitative number. Okay, so here we learned, yes, vinculin is, tailin is a protein that is under tension. It's regulated by vinculin binding, myosin activity. The important take-home message from that paper was we thought tailin is a molecule that does not only see mechanical forces, but it's also a protein that is very important for the cell, because we thought at the time it is important for rigidity sensing. In other words, it's important for the cell to know whether the substrate it's sitting on is a stiff substrate or a soft substrate. Okay, since this is all published, I will just conclude this part. I want to go to the intermediate filament aspect of it. So here we can just conclude, yes, tailin is an actin-associated protein. It's sitting in the cell adhesion sites, and it is under mechanical forces of around 7 to 10 piconewton. It's vinculin dependent, so vinculin binding to the tailin protein actually increases the tension. There are two tailin isoforms. I quickly mentioned that, tailin 1, tailin 2. It's depending on the expressed isoform. And it's important for rigidity sensing of the cell, right? So we also looked at other actin-associated structures. And if I would summarize all of the experiments we've done over the years, then I would say that many of these actin-associated proteins that have a linking activity like tailin, like vinculin, these often bear these piconewton forces like regularly. When the cell adheres, these proteins are in the cell adhesion site. And some of these proteins, of course, not all, there the quantitative experiments help you to understand that. Not all experiments will be under force. It's just not how it works, but some of them will be, 30–40% of these proteins will be under mechanical tension. And that tension, how much the protein is under tension, how many of these molecules are under tension, that is subject to regulation, right? Okay, so now as promised, our attempts to also learn something about structures that connect to the intermediate filaments, and these are the desmosomes. And we were interested in these structures because conceptually, desmosomes are very similar to focal adhesions that I just talked about, and especially the protein that we focused on, which is desmoplakin. Because desmoplakin is a protein that you can see in green that has a similar linking function than as compared to TAVI. So it can engage to the receptors, desmocholine, desmoglein, and then it can hook up to the keratin intermediate filament network. So and it has also this linker function, on the one hand association with the receptors, on the other hand association with the intermediate filaments. And this was a project that, as you will see in a second, was originally conducted in 2 labs from Alex Dunn and our group. Alex worked on desmoplakin 2 and desmoplakin 1, we worked on desmoplakin 2. And we first made sure that when we make these biosensors again, when we insert the tension sensor into the desmoplakin proteins, into these 2 isoforms, that the desmoplakins are functional. We used different assays for that, including electron microscopy. So our conclusion was, if you take a desmoplakin knockout cell and replace or we reconstitute the cells with a desmoplakin biosensor carrying this tension sensor module, then these cells are functional. Okay, so and when we started making these experiments, then that was a little bit disappointing. Our expectation was, intermediate filaments are there for withstanding force, and so we expected huge responses, but actually we couldn't find them. Actually, we spent probably 2–3 years trying to tease out any Fred differences, and if we found them, we controlled them, but never really substantial was to be observed in our cells using these biosensors. So our conclusion was, at the time, somehow it seems like these desmosomes are not under tension. When we look at these cells under homeostatic condition, we cannot see any tension on desmoplakin. And around that time, I met Alex, who visited Munich. I was still in Munich at the time, and I'm driving into the car to the airport, and he says he's also working on desmoplakin, and he can also not see anything, right? We worked on desmoplakin 2, he worked on desmoplakin 1, he looked in cell migration and so on, and he also could not see any reliable, strong difference between his controls and his tension sensor, because you always need to control that you compare your tension sensor with. And so at the time we thought, that's a little bit odd, but since we both find the same thing, probably the conclusion should be, there is no force. And we always thought we do something wrong. And I should insert here again the notion that these experiments need to be really carefully controlled. So we can really control our ass off, if you excuse me for saying so. So this is just supplementary figure 3. I don't want to go into this, just want to show it. For example, we did to find out that we really understand our experiments. So a lot of different constructs, a lot of different controls to distinguish conformation effects from actually force-specific effects, to exclude changes on pH and all these things. And so after all of this, we concluded, there is no tension on desmoplakin. And that was a little odd, but then Alex and our labs worked together. We decided we should just collaborate, and that was actually a fantastic collaboration, because Alex is also simply just a great guy, so I really like him, and it worked out just fine. The rationale then was, probably we should not analyze cells under these homeostatic conditions, right? Probably this is different from actin-based structures where you can see tension all the time being loaded on these molecules. Probably in this case, we have to pull on these cells. And this is exactly what we did. Here in the middle picture, you see the pull picture. So we had monolayers of these cells, and we took a small microneedle that you see here in this middle picture, in the upper middle picture a little bit. And we pulled on neighboring cells and then measured the Fred change in these cell adhesions in these desmosomes. And to cut a quite long story short, this is the upshot of what we found. Yes, desmoplakin is under tension when you pull on the cells. And the tension depends on two things. Here's the first one. It depends on the degree of how much you pull. So on the stretch magnitude, you could say on the external stress. Here again, you have three pictures. The middle one is a pull picture. And you see when you reach a certain threshold, then the tension sensor, which is this yellow brownish line, it decreases and the control is unaffected. And then you see desmoplakin force. And before or after, you don't see much of a change. The second thing that the desmoplakin tension depends on is the angle of how you pull. So if you have a junction and you pull perpendicular to it, you get a huge response. That is a 90-degree response here in our graph here. And when you pull perpendicular parallel to it, you hardly get any response. So we think it depends on these two things: stretch magnitude and stretch angle. And this was all done in keratinocytes, so in skin cells. Okay, so if I summarize that, then you see already it's a little bit different from the actin-based structures. So desmoplakin, in our case, in these cells, was not exposed to significant forces. We used very soft biosensors, but we also used different biosensors. But under our conditions, no tension whatsoever. But when we pull on these cells, then we see that these molecules become loaded, become exposed to mechanical tension. And the degree of how much that tension is depending on the magnitude and the orientation of the external stress. This is also to some degree desmoplakin isoform dependent. I will not really talk much about it, but if you try to bring this together with our experiments before, you could conclude focal adhesions and desmosomes, and these cells make both, of course. They make focal adhesions and matrix adhesions, and these desmosomes, they have both mechanical functions, but they're different, but probably complementary. So you need both if you want to have a functional epithelium, and these adhesion structures that on the first look, of how they are built, seem to be quite similar. They're actually doing mechanically different things. Okay, before I leave the tension sensors, still some remarks, because I always the technique is already quite established, and I see many people use it, but I'm often not so happy with how these tension sensors are used, because I think people underestimate how complex these experiments are. So that's why I'm still writing it here. It's a complex experiment. You need to control it very carefully. You have to make sure that the protein that you target is functional, that whatever you measure doesn't only give you interesting Fred changes, but that you also still measure a physiologically relevant process, right? These are not the same things. You have to pay great attention that the Fred efficiencies that you quantify are, this sentence makes no sense whatsoever that I wrote there, but just make sure that the Fred efficiencies is something that you understand and that they are specific to forces and not to something else. So in summary, all this needs to be very well controlled. And there are some studies where I think that there are not sufficient controls, and you should be just very careful about these studies, right? I mean, some of them are great that are out there, and some of them I think are could be benefiting from more controls. We wrote reviews about this, and well, evidently nobody reads these reviews, or at least not enough. Good. Let's come to the next point and leave the tension sensors, because now with these experiments we just established that there are proteins that are under tension. And if you have proteins that you are interested in, you could use this technique to find out more about this. But what bothers us then after a while was, it would be great to have a technique that actually allows us to evaluate whether these linkages that we measure are actually also relevant for any physiological or pathophysiological process, right? And as I said before, just because you have a tension on your molecule doesn't mean that this is important for a specific process. You can guess it, and you always end up in your discussion speculating. But we wanted to have a technique that directly shows us what happens if I lose that linkage that I just investigated with my tension sensor. And here we come to optomechanics, yeah, where we took kind of the same idea and just put some optogenetics in there. The idea was, and I should acknowledge the people because this was just recently done, it was really an effort by our students, and then the project was taken over by our PhD student. And I think some of you met Tanmay actually, who was on a conference in Barcelona. And sorry, Nuria, this is where I have Barcelona from. Yeah. So, but so he had this idea that, well, we could actually try to break these linkages that we are analyzing by light. Yeah, so the idea was here in the number 1, stable in the dark, we need, let's say we take a protein like taline that binds integrin on the one side and connects integrins with the second domain to the actin cytoskeleton. If we split this protein up and if we glue it together with an optogenetic dimer, and most dimers are actually can be induced to bind with light, we needed something else. We needed the opposite that can be broken by light. So we thought maybe we can use an optogenetic dimer that is binding in the dark, and then when we switch on the light, it actually breaks, and we can see right away what happens when we lose that linkage, because it's mechanically split then by light. And for us, it was also important to have this reversible, that we switch off the light, the 2 dimer halves can find each other again and reconstitute that linkage. And that is, again, because we wanted to be genetically encoded. Similar attempts have been made with chemical compounds, of course, that can be broken by light, but these are not reversible. Okay, so we found this, of course. So the optogenetic dimer was developed by Klaus Hahn originally. It's called Love Trap, and it's shown here. It consists of an ASL love domain that binds a small peptide called ZK2. And well, we regenerated this dimer and put 2 fluorophores on it so that we can see where our proteins are. And the idea was, well, if it's dark, then these 2 proteins bind to one another and we have the dimer. And if we shine light on it, and that light that's 480 light or 458 nanometer light, then we can break that linkage. Now, coming back to understanding what you're doing, the first concern that we had to look at was, if we have such a dimer, the question is, of course, if you put the dimer into proteins that are under tension, how do we know that the proteins are not breaking just because the protein is pulled on, right? So we had to make sure that this dimer can withstand the forces that we expect to have across our proteins. And of course, we know how much that force is. It's around 10 piconewton, right? So these dimers should keep binding even if we put like 10 piconewton forces on it. So this is what we wanted to test. We got into contact with our most liked physicist, Matthias Rief, and his great student, Katharina, because they are really good at this force spectroscopy experiments where you can pull on individual molecules and ask at which forces do these proteins break. And here we made an artificial construct actually, so that we can hook it up to their optical trap that they use. And with this trap, we could, we put a linker in between so that we can make the experiment repeatedly. Of course, in the real protein, the linker is not there. But through this experiment, we could have these proteins bind, and then we pull with a force spectrometer and ask how much force do we need in order to disrupt the dimer. And what we found was actually very nice, because as you can see here from the blue force, you can pull up to 27 piconewton and the dimer is still stably bound. And at around 27, 28 piconewton, it actually breaks, this dimer. So if you pull with very high forces, it breaks, as you would expect. But the forces that we think we have at these forces is stable. We also did an experiment where we used the forces that we think we have in our cells and actually pulled on this dimer for a long time. So here, many minutes actually, and you can see that in the worst case, the dimer was still bound for 2 minutes. So even like you probably have it in a cell, if you have your protein that is pulled on for quite some time, this dimer should be stable. If you switch on the light, it breaks, right? Okay. So this was a pre-experiment. We think, yes, this optomechanics module, as we call it now, is a module that you can, similarly like your tension sensor, insert into your protein of interest and then use light to break it. and with that break your linkage and then measure what happens to the cell when you do that. Okay, so here again, we did it in Taline and desmoplakin. I show you the Taline data first. You see, first we ask, can this construct actually reconstitute our Taline knockout cells? Yeah, here you see again the Taline knockout cells on the left, D, and in E, this is how the cells ideally look when you have a full-length intact Taline re-expressed in these cells. You see the cells can spread, make focal adhesions. That's how it should look like. And if you take just 1/2 of the Taline, I told you the Taline is now split up. You have the Taline head with AS-LAF domain, as shown above, or you have the second construct, the Taline rod domain with the ZTK peptide. You individually express that. Cells cannot be rescued. They cannot really spread. They spread a little bit in case of the Taline head because they can activate integrins, but they cannot form focal adhesions, cannot recruit vinculin. So that is very poor. But if you take both constructs together and you co-express them, well, then the cells can spread, they can form focal adhesions, and we get a spreading cell. So in principle, that fits to our force spectroscopy calibration, that this Taline, when it's bound, these two halves, can be like a real Taline and resist mechanical forces as the real wild-type molecule. So yeah, again, can make this short to show you what happens when you have such a, so now I have to find this here. So on the left, you see the light and sensitive control. This is a Taline knockout cell expressing both constructs, the Taline head, the Taline rod, and it's connected by these two, by this optogenetical dimer. That's why you see focal adhesions here. But the AS-LAF domain is mutated and it's light insensitive. And if you look at these cells, then you basically can see that nothing happens. The cell is just moving around a little bit, but not much is happening. On the right, you see again a Taline knockout cell that expresses now the real optomechanics construct, a head domain and a tail domain that is carrying this optogenetic dimer. And you see immediately what happens when you switch light on it, the cell is collapsing. You see here actually that there are some adhesion structures are left behind. So the effect is so strong that actually some of the Taline and actually the adhesion molecules that anchor the cell to the substrate are ripped out of the cell, and the cell is in many cases severely collapsing when you break the Taline linkage. So this is interesting and somehow pleasing to see. The Taline linkage is indeed not only there and bears forces, it's also really important for the cell. Yeah? And I told you that in focal adhesions, you have many molecules that are potentially important for this force transmitting process. But in fact, if you lose Taline, you have a big problem. Yeah. And of course, we knew that Taline is important, but here you acutely take it away. The cell has no time to compensate in any way. And so you see what the function of the molecule is. So Taline is really important. You can, of course, quantify this. Yeah, then you get these numbers. We did this with many cells. So the Taline linkage is really essential for cell adhesion maintenance, at least in our cells. Yeah? So the loss of the Taline linkage in these cells cannot be compensated. And yeah, okay, you can do this, and you can take individual adhesions, and when you know this dashed area always indicates where we blitzed the cell, so where we optically stimulated the cell. You see individual focal adhesions will also collapse when you blitz them. And here you have a cell. What happens when you just shine light on half of the cell? Well, that part of the cell that you are irradiating will collapse. So this gives you an idea of how important Taline is. Wherever you illuminate the area and you break the Taline linkage, that area will also collapse. Okay, also this can be quantified. Let's go to the desmosomes. Yeah, because of course we thought, well, for Taline, we now find that Taline is important. What about the desmosomes? Here, I remind you of what I just told you 5 minutes ago. Desmoplakin is a molecule that is a protein that sees mechanical forces as well when you pull on these cells, right? And so we wanted to do the same thing, and that's what I can show you now. This is, again, here I should show you this. So we split up the desmoplakin 2 protein into 2 parts. And actually where we before had inserted the tension sensor, now we inserted this optogenetic optomechanics module. So that we have an N-terminal fragment, a C-terminal fragment. C-terminal fragment in this case binds to keratin. N-terminal fragment hooks up to the desmosomal structure and should make the linkage. Also here we used electron microscopy to make sure that we have actually desmosomes forming in these desmoplakin optogenetic constructs. Well, and here's a movie that shows you what happens when you actually blitz the cell. And as you can see nothing, right? So this cell here, this is in fact a movie. In the middle, you see these desmosomes. So these are again desmoplakin null cells, knockout cells that are reconstituted with our desmoplakin optomechanics construct. So under these conditions, absolutely nothing happens. Maybe some of you keep thinking, are still with me. This is not surprising because we measured before that under homeostatic conditions, desmoplakin is not under tension, right? So what happens when you actually pull on these structures? So these are now desmoplakin null cells reconstituted with the desmoplakin optomechanics construct. We pull on the cell and at the same time we blitz them here in this dashed area. And this is what you can see. Yeah, when you pull on the cell and you irradiate this area, then the junctions actually break. Yeah, that tells you that desmoplakin is actually very important for maintaining this structure, but only when you pull on the cell. And the control is shown next to it. This is the light insensitive control. Again, we take knockout cells, re-express the desmoplakin light insensitive control, pull on it, as you can see here, irradiate this area, but actually it doesn't fall apart. You could even think that it's reinforcing a little bit. Okay, so conclusion from these experiments, and this is basically the end of my talk, the desmoplakin linkage, it is essential for cell adhesion when external stress is applied. And since we always try to make evidence for everything that we want to state, I think that was good to see that these experiments, that there's a functional role here, which of course fits very well to what is known already. That desmoplakin knockout mice are of course known for a long time that they have a severe phenotype and that they are patients with dysfunctional desmoplakin. It fits this phenotype, right? These patients, these mice get a problem if their skin, for example, is exposed to high external stresses. So in summary, I want to make this very short, just 3 points. So the tension sensors can answer the question for you, does a particular molecule experience mechanical tension? I told you a lot about that this is not a trivial experiment, but you can do it. Yeah, the optomechanics approach can go one step beyond, we would say, and answer the question, what's the consequence of acutely losing a particular molecular linkage, maybe a linkage that you measured before with your tension sensor. And well, what does it mean for the biology that I showed you? Well, obviously the actin and IF associated structures differ mechanically. That's maybe not a surprise. I think the actin-based structures are understood quite well. My feeling is, but that's maybe just because I'm not an expert, you can tell me better, but I think we still have to invest quite some work into the IF associated structures. We have to understand better how the different IF networks contribute to force transmission process or not. So I think, at least to my understanding, there's still a lot to be learned, and maybe that's why these tools that I presented to you today are beneficial to some of you. At the end, I would like to acknowledge the people that did it. So yeah, Tanmay, I already pointed out, who I showed, and I have to point out the 2 most important people, the 2 girls that actually run the whole show, and I'm just giving talks and doing this sort of things. So Ina and Anna are keeping everything rolling. And then of course I should acknowledge a lot of the collaborators that we worked over the years with. Matthias is the most important one when it comes to force spectroscopy. Many others too. I saw Thomas actually in the audience at the beginning, so he told us how to work with these keratinocytes. So a lot of credit also to him. And yeah, if you're interested in these tools, don't hesitate to ask me. If you're interested to work on such a project in our lab, also don't hesitate to ask me, because we are looking for PhD students. Okay, thank you very much. That's what I wanted to tell you, and I'm happy to take questions.